Studies of stable isotopes during the last decades become one of the fastest developing fields relevant for the whole archaeology, because they allow reconstruction not only of the diet but also of the human mobility and past environments. Studies of stable isotopes are important for the reconstruction of the past diets, and the fact that they are stable, so they do not decay in bones over time, make them suitable for the investigations. The first step in the isotopic study is extraction of collagen, and after that material is analyzed by mass spectrometry in order to detect amount of specific isotopes. Those amounts may indicate what was the diet of an individual, because different foods have different isotopic signals. As it illustrated on the picture A, stable isotopes such as carbon-13 or nitrogen-15 are used often because those isotopes may distinguish between intake of marine and terrestrial animals, or may detect diet based on specific type of plants such as maize or millet. What is recently discovered and have huge potential for the study of diets is that isotopes analyzed in different tissues represent the diet in different periods of life. For example, if some child was fed with marine food until age of 5 and then changed the diet to terrestrial food, different isotopic signals will be found in different dental tissues. Dental tissue is type of tissue which do not remodel and kept the dietary signal without changes, and consequently kept the dietary signal of the moment in life when this part of teeth was developed. In the contrast to the dental tissue, postcranial bones are remodeling, and that is why that part of skeleton record the intake during the few last years of life. With carefully planned multi-sampling strategy of different dental and postcranial tissue, and the procedure during multi-sampling strategy you can see on the picture B, may provide us possibility to investigate dietary changes during life. And when this approach is applied in children, it is possible to sample dental tissue formed during fetal development, which presents the diet of the mother during pregnancy. When sampling other dental tissues, it is possible to detect the duration of the period when only mother's milk was consumed, which is actually duration of breastfeeding, and also to detect the time when some new food, weaning food, was introduced into children's diet, and also to analyze isotopes to detect what was the weaning food which was used for the feeding of the baby. This multi-sampling strategy make possible for us to detect what the mother of the child who died at the age of 4 and was buried at the Lepenski around 6000 BC was eating during the pregnancy, and we estimate that her nutrition during pregnancy was based mostly on aquatic food, the same as nutrition of the mother of the newborn baby who was buried at the Lepenski at the same time. This 4-year-old child was breastfed until first age of life, and because we analyze other children from the Danube gorges, that make possible to conclude that this duration of breastfeeding was much shorter than duration of breastfeeding of some Mesolithic, but also of some Neolithic children in the same region. Also, that give us opportunity to investigate what was the weaning food which was given to him when breastfeeding stopped, and we concluded that weaning food was made from some kind of cereals, which was a very new type of weaning food in the region of the Danube gorges, because most of the Mesolithic, but also many Neolithic children were at that time still weaned with aquatic weaning food.